Hello, everyone, and welcome back to the Demystifying Medicine McMaster podcast. Today, we will be touching upon the topic of rare lung diseases, specifically idiopathic pulmonary fibrosis. This disease has an estimated prevalence of 13 to 20 per 100,000 people worldwide. Unfortunately, there's no cure. Dr. Naftali Kaminski, professor at the Yale School of Medicine, is one of the researchers that has been advancing our understanding of the molecular and cellular basis of the disease. Using single-cell RNA sequencing, his laboratory has built an IPF Cell Atlas that is helping define disease-specific transcriptional profiles and functions in disease pathophysiology. So I guess we'll just get started then? Okay, so it's amazing that research in IPF is increasing, especially with your role in creating a platform that allows the public to conduct their own research by using the data set from Cell Atlas that you've recently created. So what was the process like in creating this project, specifically sequencing the RNA of more than 300,000 cells from lungs affected with conditions of a chronic lung disease?
So the question of process is complex because research projects have sort of three or more layers, right? The first one is conceiving it. You start thinking what you want to do, create the concept. And I think when the first reports on single-cell technologies came out, the promise was obvious to me and to Dr. Van Rosas, who at the time was at Brigham and Women's. So we start discussing potential collaborations. And the question is, and I'm not sure that that's what you're asking, but I'll still describe it, is that what we needed to understand is, can we do it? So what are the technological things that are needed to be done? And then how do we do it? So how do we obtain the funding? And so there's certain problems with single-cell technology. Single-cell technology is basically a droplet-based technology, at least the technology we thought of using. Basically, you need the cells meeting with being barcoded, and then a cell-specific library is generated. And for that, you need the cells relatively intact. Cells that die don't have less RNA or are degrading or have intact membranes. Usually will have different signals. So the technology, which is extremely simple to do, very straightforward kit work when you're doing like cell culture work, because you're taking live cells, you're mixing them with a bunch of reagents and basically slipping them in the machine, in which what happens is by microfluidics technology, every cell meets a droplet with the barcode. All the RNA in this cell gets barcoded, and then there is a mixture of all the cells that goes back into the sequencing. But now when you're sequencing, we know that some origin of every RNA species, and then computationally we can rebuild the cells. This is very hard when you're starting to deal with human tissue or with tissue or with organs, because an organ, you have the cells sitting in their extracellular matrix, in their microenvironment. You need to dissociate the organ. The process of dissociation is actually hard for the cells. You may break down the membranes, you may kill some RNAs. So we had to work out all these conditions. And Van Rosas was really instrumental in developing methods to both obtain human tissues, store them in a way that allows maximum amount of cell viability. The question of why do you need to store them, why can't you just take the lung and dissociate it and run to the machine, is the tissues we get are mostly from lung transplant or from surgery. They come in different times of the day and the night. there may be some differences in difficulty of surgery or timeliness or other things. So if you do every sample on its own, what you get is basically that every sample has its own what we call batch effect. So effect of the unique processing. And what this creates is a huge difficulty in analyzing the data because it's really hard to distinguish between what is really the individual signal towards the technical signal of differential handling. So what we wanted is a standard process in which every sample is handled the same way, goes into the storage, and then when we bring the samples, we bring them in large groups, 8, 16, 24. We analyze them together. And now at least the batch is not individual, it's for a grouping of so all of these things needed to be solved, thought about it. And actually, if you look at our data, one of the coolest things is there's really very little batch effect. So whatever we did worked. There's issues with dead cells and other things, and we can discuss. But there's a lot of technical things that we actually detail in the supplement of the paper that needed to work out. So probably to get to the stage that we could do the experiments, I think was a joint work of at least a year of calibration, testing, developing. Much of what we know of the lung cellular distribution comes from a very small number of individuals. The other thing is that there's a paper we published a few months earlier in Science Advances, which was led by Sam Maradon and Laura Nicholson's work group, in which we looked at the normal lung connectome. And in that paper, we looked at what are the conserved networks, connectomic networks between mammalians. So we did rat, mouse, pig, and human. And this is also, if you look at it, it's also one of the largest collections of normal lungs in general, mammalians. So together, this sort of allows us to start understanding what is the blueprint of the lung, the adult lung, and start just understanding how things go on. When you look at the whole cell atlas and compendium, because remember in our website at www.ipfcellatlas.com, we have actually other data sets, not only ours. So there's the data set from John Kropsky and Nick Vanovich, and Sasha Misharin and Bob Lofiatis. So in general, although they're smaller, actually the Misharin is the Kropsky Vanovich is relatively large. Together, we have over 500,000 cells, and you have multiple interstitial lung diseases. So again, you're starting, although they were done differently, all these data sets are available online, and they're all in our website as for data query. So now, regardless what disease you study, you can start comparing to all of this tech. And I think that's going to have a lot of importance.
In your recent study published called Single Cell RNA Sequencing Revealing Ectopic Aberrant Lung Resident in Cell Populations in IPF, you highlighted the discovery of aberrant vasaloid cells. Could you please explain what is unique about these cells? Also, in this study, it was found that there was a small sample of COPD patients which also had these cells. And then do you think that these cells can be found in other lung diseases?
So these are multiple questions and sort of multiple tenets. So I need to unpack this question a little bit. So one is the question of what's in the name, right? So how do we actually name cells? And the history is relatively simple, right? Initially, people named cells by what they saw, right? Macrophages, they're big eaters, right? And then during history, we started describing cells by their shapes, by their location. All the names of the cells we have are sort of in some ways, a combination of an arbitrary location and a system. And there's always arguments like the famous, there's some cells in the lung that change their name, like R cells and some cells. And then there's definitions like markers, the way immunologists do, right? So they go by sets of markers. When you look at this big data set, you suddenly look at, thousands and thousands of, hundreds of thousands of transcripts, actually millions transcripts from hundreds of thousand cells, and you start classifying the cells. What you do is basically, the first thing you do is you cluster the signal. And that generates these beautiful images that we have in the paper. And when you cluster the signal, now you start looking to say, okay, is this cluster a specific cell type? And you start looking and seeing, does it fit cells that you already know? And over the years, beyond the morphological and topographical definition of cells, we've gotten also some molecular markers. So we know that certain molecules like surfactant and others are not expressed in most lung cells, but are expressed in type 2 cells, alveolar type 2 cells. But think about this, even this nomenclature, alveolar type 1 cell and alveolar type 2 cell, almost arbitrary, not functional. So in the purpose of analyzing the data, you classify all the cells into specific groupings. And when we looked at the clustering, there was one cluster of epithelial cells that didn't show up in one. And if you look at the paper, you can see these sort of almost this small dendril. It's very obvious. And these cells were really unique. And they were unique in a way that actually got us really excited because they were definitely epithelial cells. They expressed ectom and other things. They definitely had some, how would you say it, some senescent characteristic. They expressed markers of senescence like P16, P21, PDF15. They expressed some markers of airway basal cells, the sort of progenitor cells, but they didn't express all of them. And that's why, so it started becoming obvious that they're not a regular cell type. Now, other people have been doing really cool work in the last year and in parallel, which is even cooler, to show what is the source of these cells. Who are they? Are they alveolar in general? Are they airway? Are they progenitor? But for us, what was exciting is this sort of aberrant nature of these cells. They also had mesenchymal markers. So now you have a cell type that has both an epithelial and a mesenchymal marker. This is well known in cancer. This is well known in development. In pulmonary fibrosis, it's been shown probably around 10 or 12 years ago. But then subsequent work, which was based basically on lineage markers in animals, in mouse work, claimed that these cells didn't exist, which was sort of weird because all of us actually saw them. And the other interesting thing that these cells have also some progenitor capacity. They carry some transcriptional. So now you have a cell that's both epithelial, both mesenchymal, is a little bit old, or senescent, or rogue, you say, a little bit progenitor. And then there was one more thing that I thought was really cool about these cells. They actually carried some of the most prominent pulmonary fibrosis markers we found. So for instance, I started my career around actually close to 20 years ago. At the end of our postdoc, this discovering that a certain matrix metalloprotease materializing MMP7 was increased in pulmonary fibrosis. I did it by the most cutting technology that was then microarrays, the most cutting edge technology. And we measured, I think, 600, well, actually, started with 588 genes at the same time and ended up with 6,000. This was a big deal. And in this paper, one of the most increased genes in the IPF lung, and this was like ridiculous. I did 4 IPF and 5 IPF and 3 controls, whatever. And materializing was the most upregulated gene. At the time, it was a big deal. The common dogma in pulmonary fibrosis was that matrix metalloproteases were decreased and inhibited. And we did some work, and this was published in PNAS, showing that materializing knockouts on 2 backgrounds were relatively protected from fibrosis. I sort of decided that my career will not be on MMP7 as an early career. And again, for you students, the thing is you make decisions, but you don't really control the fate. And but MMP7 kept popping up again and again and again. Actually, in my first screen of the peripheral blood proteins in pulmonary fibrosis, a limited proteomic approach, I think we looked at 100 proteins. MMP7 distinguished patients with IPF from other diseases. And now we look at the gene expression, and suddenly MMP7 pops up. And actually, this is the cell type that MMP7 is interested in. So again, an interesting story, a weird type of cell that starts to answer questions we didn't even know we were asking. And then I look at the data and I see another thing, which also relates to my career, because I trained with Dean Shepherd, who discovered that alpha V beta 6 is an integrin that activates TGF beta and drives fibrosis, and is now considered a drug target. And then I look at the lung. These cells express the highest level of alpha V beta 6. So these are progenitor cells, look like defective basal cells, not fully differentiated, or maybe reverting, with a little bit of senescence, epithelial mesenchymal characteristics, and known markers of fibrosis, and they can activate the key molecule that drives fibrosis, TGF-beta. So they must be important. So we did the stain, and these cells show up at the hallmark of the fibrotic lesion, the myofibroblast foci, and they sit at the edge. These are the cells we've, for years, many of us, and I want to highlight definitely Carlosi, an Italian pathologist, that pointed it out really 15 years ago. People used to say that these cells are just flattened type 2 cells, so hyperplastic alveolar type 2 cells. But he said, no, these cells have activation of other programs. And he actually called them I don't remember exactly, but this like really groundbreaking paper. And he was right. At the edge of myofibroblast foci, there's cells that are different than any cell in the lung and represent probably either a block in differentiation or a loss of differentiation, but definitely a recapitulation of the developmental program, which actually is the title of a paper I wrote 20 years ago with Moises and Annie Pardo. And so when you think about it, so you find them in IPF lung, in almost every IPF. They're not that common, but the signal is very strong. These cells are very active. We did find them in several COPD lines. Very few cells and very few, but they don't localize to the lung parenchyma. And we didn't find them in normals. When we looked at other data sets, they showed up in the fibrotic data. I think they showed up also in non-IPF tissues, but definitely in fibrotic lungs, and they showed up. We didn't do the staining, so we don't know if they showed up in the parenchyma. But I would think so. And what I would think is that these cells probably, and based on their transcriptional program, the sort of SOX911 signaling and other things, these cells probably show up whenever the lungs needs to rebuild. And they probably are not alveolar, they're not distal lung. They're probably related more to the airway. And the way you want to think about it, what I'm saying now is probably could be completely wrong, but it's very appealing to me, is that there is an order to building the lung. You know, you look at development, the lungs starts with a tube. When the tube is sort of relatively complete, it starts branching. And after it branches, it starts having these vesicles, the alveoli. So that's the way the lung. There's no process of really building an alveolus. I think so. So I think what's happening in the lung because of, or there's local repair mechanism. So what I think is happening and probably these cells are important in the rebuilding stage, probably relatively late. And what you see, these cells represent the sort of the building, they sort of failed effort of building it. But again, I may be wrong. So we will be standing for them in COPD soon, and we'll see. But I don't think we'll see them in the lung parenchyma the way you see them now. Actually, it's not a novel cell type because the cell has existed. It's exactly like Columbus does not discover America; it was there before. So when you think about the endothelium, we didn't know that there was this subgroup. So when we looked at the data, and this was a really, there's several people that did the work. Taylor Adams, who's actually a person who is in between college and and grad studies, and has done more work, more innovative work than all of us that have doctoral degrees in the team. And Jonas Schoep, who is a physician that never had any computational training before, who did amazing work in sort of outlining and analyzing the data. And then Jonas was insisting that he's seeing an endothelial cell that is not typical in the lung parenchyma. And when we looked at it, there's definitely a subset of endothelial cells that carry collagen 15. You don't really see them in the lung parenchyma in normal lungs or in COPD, but they show up in IPF in the lung parenchyma. And when you stain, you see them in areas of dense fibrosis. There was a group that published sort of an airway Cell Atlas from in Nature Medicine before us. So we contacted them. They were really helpful to share the data with us. And what we were able to show that these cells usually are part of the peribronchial vasculature, but not the alveolar vasculature of the epithelium. So these cells are basically, we call them ectopic because they were not in the right place. Now, why is this important? I don't really know. It may support, again, the idea that the lung is trying to rebuild airways mistakenly instead of building alveoli. But it also may reflect an important thing about symptomatology of the disease, because we tend to think a lot about the fibrosis, the scarring, and the restriction that patients with pulmonary fibrosis experience. But another really important feature of the disease is actually the loss of diffusion capacity, basically the loss of gas exchange surface. In IPF, there's a decline in alveolar type 1 cells, which are the cells that carry out gas exchange. But there is a possibility that also, if you are replacing the endothelial cells that are supposed to be in alveolar, that are designed for gas exchange, with these weird endothelial cells that are built for more for blood delivery than for gas exchange, maybe you also reduce the diffusion capacity. Therapeutically, it's hard to know. We're just starting to study this. This actually got us thinking about the fact that probably there is a need to describe all endothelial cells in the human lung, um, but this is future.
And then with these new discoveries, do you think that being able to detect the aberrant vasaloid cells or monitor the vascular endothelium in patients could be a viable and effective method for early diagnosis in IPF patients?
So yes and no. So no is because if you need to find these cells by getting a biopsy, there's no way we can do it. There's no clinical justification to do biopsies on people with early disease. The risk is too high. On the other hand, if we develop other non-invasive technologies, um, we could potentially develop imaging technologies that will allow us to see sort of if somebody, there's a common phenomenon that occurs in people that called interstitial lung anomalies. And if some of these are nothing, some of these are just markers of viral infection, some of these can be markers of future pulmonary fibrosis. Um, so if you show that a set of markers of these cells is there, probably that's abnormal, but this is really a great area for research for the future. The second is actually more straightforward. Now that we know that, for instance, the source of MMP7 in the lung is mostly from these cells, maybe, and we know that MMP7 is increased in the blood of people with IPF. Maybe if, and I'm not saying MMP7 specifically, it's also GDF15 and other markers. Maybe if we identify secreted molecules that are really specific to these cells, then we could use this as sort of peripheral blood markers for early detection. It's a lot of work. The signals we have now are really the edge of the iceberg, right? So we need more lungs and potentially proteomic analysis of these cells and metabolomic analysis and other things that will allow us to identify secreted. But the cool thing that now we can say, what is the specificity of the marker? I'll give you another examples. The profibrotic macrophage is described by Sasha Misharin and by Bob Lofiatis' group. Is also they have characteristics, MMP9, YKL40, Ostoponte, and other molecules. So in theory, the same thing. We could look, do you have profibrotic macrophages in your lung? We don't know, but let's look at your peripheral blood. And I want to highlight the work of one of the best markers that we have for lab outcome prediction in IPF is really simple. It's the number of the monocyte count in the peripheral blood of IPF. And this was a beautiful work of an MD PhD student, Madeline Scott, that was published in the Lancet Respiratory Medicine, in which she basically reanalyzed our data and other cohorts and basically showed that that's been replicated. So information derived from genomic data sets is really useful, but sometimes it's not useful in the direct way you think. Sometimes it becomes even something. So I think looking at these cells, having a better signal. I don't know, for instance, if we can find DNA of these cells in the peripheral blood, plasma-free DNA. But yeah, I think that finding evidence for the existence of these cells in minimally invasive methods could be useful for early detection.
So, moving on to the topic of the current pandemic and the coronavirus, we know that the symptoms associated with COVID-19 are diverse, ranging from mild upper respiratory tract symptoms to severe acute respiratory distress syndrome. So, the major risk factors for severe COVID-19 are shared with idiopathic pulmonary fibrosis, namely increasing age and the comorbidities such as hypertension and diabetes. With your understanding, Do you think that the same biomarkers that you've discussed in your previous research will be prominent in revealing the pathophysiology of IPF patients who have contracted COVID-19?
That's complicated. We don't really know much about IPF and COVID-19. Thank goodness our patients sheltered in place and were careful, and so very few were affected. COVID-19 is basically is a viral infection in which the majority of the damage to the lung is caused by inflammation and its abnormalness. We just posted the paper in Biomed Archive looking at single-cell analysis of the peripheral blood of patients with COVID-19 and progressive. And what we find is a dysynchrony between innate and adaptive immunity, which may cause both a hyperimmune response and immune paralysis-like response that together will both cause a lot of damage, but also impair the clearance of the virus. I don't really know yet that there's an overlap between these techniques. I actually don't think that that IPF is really has a strong relation to COVID-19. There is a relationship between post-viral disease and severe viral pneumonia and lung fibrosis. That's been shown for SARS and MERS. It's not, the data is not strong. We don't know yet about COVID-19, but my guess would this would be in different mechanisms and potentially a different type of disease, which is more of the development of post-inflammatory scars that don't really progress. Having said that, we don't know because, if you think about the gene variants that predispose to pulmonary fibrosis, some of them, like McMaster, are really common. And some of them are relatively rare, but even the rare ones are common enough in the population that if you have 5 or 6 or 10 million people in the world affected, there's a good chance that, let's say, a few thousand of people that have a predisposition for IPF would be infected. And we don't even know what's the interaction between a severe viral infection and a gene variant that predisposes to disease. I suspect that many of the variable reactions to COVID-19 are actually a result of these rare gene variants. So if you think about the hypercoagulable states, gene variants that are associated with coagulation are really common in the population, if you think, but as single variants, they're not, but as a group, they're common. So the chances of a rare phenomenon exposing, a common phenomenon exposing rare manifestation of disease is really, could be with fibrosis, we don't know. There's a lot of discussion about potential fibrotic changes and other things in post-COVID-19 patients. I think we have to be really careful. We have never had the experience in the current ability to image and look at data of encountering an unknown virus. So basically, again, we have million patients, and the rare manifestation may dominate our view. So we don't really know at this stage.
Yeah, so as you mentioned, that area of research is currently there's a lot of uncertainties. So we know that currently the role of antifibrotic therapy in patients with IPF who contracted SARS-CoV-2 infection is poorly defined. And a major target for antifibrotic therapies is the transforming growth factor beta pathway. And the SARS-CoV-2 spike protein contains an arginine, glycine, and aspartate integrant binding domain, and a number of the coronaviruses contain an N-terminal galactan fold, raising the possibility that therapies that inhibit integrins or galactans might be a benefit in treating COVID-19. So through your studies of improving diagnosis and treatment for patients with IPF, do you have any insight on the advancing therapies that involve this pathway?
So the role of integrin inhibition in COVID-19 is interesting, but it's probably not the key therapeutic intervention. We have not studied it directly. It's just that I don't, that's not my research. But I've been very interested in integrins for many years. But again, I'm not an expert. There is a rationale to inhibit TGF-beta during acute lung injury. And that's because TGF-beta has an effect on alveolar permeability and alveolar variability. And there's some really cool work that was established. And COVID-19 represents an opportunity to study acute lung injury in a very organized way because of the lurking nature of the disease and the fact that it all happens at the same time. So you have a period of a few days in many of the patients in which they have mild disease, in which you can try actually to prevent acute lung injury. So many of the studies that people have tried in acute lung injury failed because the causes of acute lung injury dominated the outcome. And patients showed up with already ARDS, with full-blown disease. So here we have an opportunity to prevent. So there's a chance that inhibiting TGF-beta early, locally, may be protective. It could also rebound because it may increase inflammation. We do not know if the kind of inflammation that's increased by inhibiting TGF-beta will be pro-lung safety or against it. So again, it's a valid target. Integrins are definitely. Now, the whole binding to the spike protein, again, there's a lot of interesting things. The main thing is that, and again, I'm not an expert, but I would say that spike protein binds to ACE2 much stronger than most of it. So again, there's a lot of research that needs to be done. I think in general, COVID-19 is a tragedy and a completely preventable tragedy. But in general, it does provide opportunities for us to sort of strategize about acute lung injury. So the way I look at it is like 3 or 4 types of drug families. One is the drugs that inhibit early infection. Second is the drugs that kill the virus. Third is the drugs that prevent any hyperinflammatory symptom. And fourth is the class that people have less paid attention, which is the lung protective drugs. And I think that interventions that will potentially affect integrins may be more in the lung protective than necessarily the antiviral. I hope that answers your question.
So more recently, IPF has been thought of as being a disease of repetitive injury of the alveolar epithelial cells with aberrant wound healing resulting in fibrosis. It is understood that this disease is more frequent in males than females. In your previous study on transcriptional regulatory model of fibrosis progression in the human lung, all the subjects are being tested were males. Do you believe that there is a limitation in truly understanding the potential differences in the presentation and disease, its comorbidities between genders?
Yeah, definitely. So all of us that see IPF patients have, I would say, different gestalts of the disease, right? So there's a disease that's common in more older men. There's a disease that's common more in, how do you say, middle-aged, upper middle-aged men. There's a disease that's common in sort of slightly older women, and they look slightly different. We don't really understand. The problem is that to study it, the sex effects of a disease, you have to study it directly, right? So, because when you try to study them within the context of the whole cohort, what you see is basically the effect of selection biases, population biases, other things. So, if you look at transplantation, for instance, that's where we get most of our tissues. It's done, or used to be done more in men than in women. I don't know, I need to look at the numbers now to sort of update it. But especially in Europe, it's done more, I think, in men again. And I think part of it is basically severity of disease and rate of progression. Because if you're stable, the person gets to an end stage when they're too old to get the transplant. And there will also, historically in the US, there were also other reasons, small lungs, big lungs. Most of the lung donors are men. So there's a lot of, so I think availability of tissue, other biases. For instance, women are less likely to be diagnosed with IPF, just because people are thinking they have more autoimmunity. The breakdown actually is 1 point, I think it's 2 to 1. It's not huge problem disease in this case. But I think there's other biases. So I think actually creating a cohort in which you study the progression of IPF or of pulmonary fibrosis in men versus women respectively, with this question in mind, not as a retrospective analysis. It's often the same that I've been preaching about sarcoidosis and race effects. So we tend to do these retrospective studies or these big cohort studies and assume that things will fix themselves. But no, we actually have to put it in the question. So we need a cohort that asks, in sarcoidosis, is the disease really different in white and blacks? And then we design the study in a way that addresses all the other determinants. So the same thing with men and women. We need a cohort. In general, we need cohort studies in IPF. There's registries. These registries don't have hypotheses. They don't do balance recruitment. We need a cohort that actually looks forward. And there's some things, like for instance, is the extent of familial disease different between men and women? There's ways to look at it, but the main thing would be to do it forward. There's also been a lot of misinformation. So, for instance, many people in their training, like I used to do Blow-Meissen model of fibrosis in female mice and rats and not in male. And I don't even remember why we did this. I think the notion was that you get better fibrosis. It's not true, actually. Male mice seem to be more prone to fibrosis than female mice. But even this has not been an area of detailed investigation. It's been sort of the arbitrary observation. So I think actually the impact of sex in pulmonary fibrosis in general, and then in IPF specifically, is really important. It has to be studied, sort of put on the front line and studied directly. In tissues, I think definitely lung transplant, we've had way less women historically.
Thank you so much for taking the time to meet with us. You've really helped us get a lot of detail about IPF. Do you have anything to add?
Well, I think the most important thing for me is actually One is, of course, spreading the information, but the other thing is spreading the message that it's really not about a single group. So we made sure we were aware that Ivan Rosas and I and others were aware that there were other groups doing the Cell Atlas. And we communicated with Nick Panovich and John Kowski, and we decided to coordinate submissions. Actually, when we posted the papers on bioRxiv, we actually texted each other to coordinate the exact timing, and also we coordinated the tweeting about it. And the same thing now, when we did the co-submission and really insisted that the paper will be in the same issue. And I think the reason for this is really that pulmonary fibrosis community, although pulmonary fibrosis is a really important disease, it's still a small topic in the world. So if this community has no reason to be, because its competition is with everybody else in the world. On the other hand, our patients do need us to compete for attention and money and resources, because theirs is an unmet need. So what we need to do is, it's all about the community. And that's why I think that's why I was really excited about participating in the series and having the Demystifying Medicine and actually speaking with students, because we really need a large community of scientists, advocates, physicians, caregivers, everybody to solve this problem. And I think that many of the questions we solve in fibrosis are going to be relevant. Pulmonary fibrosis are going to be relevant to aging, to quality of life, to function. Fibrosis in itself is extremely common. We don't think about it, but when somebody has a knee injury and they can walk a little bit, that's fibrosis. And what happens to the heart after injury is fibrosis, and the brain. So understanding the basic mechanisms of fibrosis, and pulmonary fibrosis provides a relatively accessible opportunity to affect the health of many, many people and really have a huge impact. But for that, we need multiple brains working in coordination, not just a few groups. So for me, it's the collaboration, the data dissemination, and the sharing that's really exciting. And that's why, our website, the IPF Cell Atlas, the way when we built it, I thought, we don't need a website for an expert. We need it for an expert in pulmonary fibrosis, or for an expert in interstitial biology. We need a website that every scientist, every person who understands biology could look at the interesting genes. That's the way. So I think the most important thing, the most message that is important for me, we need to break out of our protected bubbles and be out there and share data and communicate about data and really be stronger together.
Thank you so much, Dr. Kaminski, for taking the time to speak with us today. So to wrap things up, hopefully through this interview with Dr. Kaminski, we have a better understanding of the current research and efforts made by healthcare professionals to revolutionize immunophenotyping in advanced lung diseases. We are continuing to be a step closer to helping patients who feel invisible and to help them find effective therapies that would help alleviate the burden of these rare lung diseases. Thank you for listening.